Good morning, everyone. As part of the ENSEMBL regulation team, I will present you today our ENSEMBL regulatory annotation pipeline that we developed and we improved for being able to identify regulatory elements in the genome of the fish species that were involved in the AQUA FAANG project. So as you know, this consortium has produced a lot of high-quality genomic data that was even recently presented to us, and all of this data is currently, or most of them, is currently easily accessible through the FAANG data portal, so it can be used not just for us, but for all the community that is interested on them. So as part of the consortium, we have used this data and we have developed a new pipeline that allows us to process all this new data generated with these new protocols and these assays, and to identify regulatory elements in 5 of the AQUA FAANG species that were having data registered until July 2023. That is the month where we finish our first step, that is the registration. Yeah. So the name that we use for, in general terms, to refer to our pipeline is the ENSEMBL regulatory build. So basically we have 3 stages. The first one is the importing or retrieving data and experiments from public repositories like the FAANG data portal or ENA or ENCODE for the case of human, because we already provide annotation for human. This data is added into our databases and is then processed by our primary analysis pipeline. This pipeline is aimed to produce high-quality data in terms of peaks and signal tracks from ATAC-seq, ChIP-seq experiments that are then used for identifying these regulatory elements in the genomic regions. I want to highlight the main changes that we were doing in our pipeline for adapting it to the AQUA FAANG data experiments. So this primary analysis pipeline involves a lot of different pipelines that are basically designed for the different types of assays that we're interested in to analyze. But all of them involve the same steps that are the ones that are summarized here and are based in the same set of tools, right? So we always start from the sequencing reads. The first step that we perform is to remove remaining adapters that can be still attached to the sequencing reads. So for doing that, we use FASTP for single-end experiments and ngmerge for parent sequencing experiments. After that, we perform the alignment of the reads by using bowtie2 with different configurations also for parent or for single-end experiments. And at that point, if we have technical replicates that came from the same biological replicates or the same organism, we merge the alignments by using some tools. So after this step, we will finish having one alignment per biological replicate for each experiment, right? After that, we perform peak calling by using GenRich, combining all the information from the different biological replicates. As you can notice, we use GenRich. This is a relatively new peak caller. We are using it because for us it provides several advantages. One of them is that it provides different parameters that we can adapt for our different pipelines. In our primary analysis pipeline, we can adapt it for ATAC-seq, ChIP-seq, DNA-seq. And we also opted for using GenRich because it can handle multiple alignments that can be originated from duplicated genomic regions, like the ones that we know that are present in the fish's genomes. And also because it allows us to combine information from different replicates before performing peak calling. So after we just need to run or to perform one peak calling task without requiring a post-processing like IDR, right? So I will try to explain in a broad way how GenRich works. So basically, it starts by taking the alignments from each experimental replicate and to creating an experimental PyLab simply by parsing the alignments to obtain fragments and then counting how many fragments we have per each genomic position, right? Per each replicate. And at that point, one interesting feature is that we can exclude some genomic regions that we know that can be masking our real peaks, right? Then it does the same for the control replicates if we provide them, as it can be done in a ChIP-seq experiment. And if we don't have control replicates, it estimates like a background level noise from the same experimental replicate. Then these values for each genomic position are combined, assuming a log normal distribution in order to obtain p-values, again, per each genomic position and per each experimental replicate. After this step, the p-values from the different replicates are combined. So at the end, we have a single p-value signal that is representing the p-value across the whole genome, right, for the experiment. This p-value, if the user specified, can be converted to a q-value signal. And then this is the one that we use for identifying regions that are reaching a statistical significance that the user specified, right? So these regions, like the ones that are highlighted here, right, are regions in the genome where this statistical significant threshold was reached. And then there is a final step where we compute, well, we know, GenRich, sorry, computes the total area under the curve for the region, and if the value is higher than a threshold that the user can also set up, then the region is called a peak, right? So as you can notice, there are different parameters that are involved here that we can use and adapt for our different pipelines, right? And one thing that I want to highlight here is the exclusion of the genomic regions. Because we were implementing improvements in our pipeline in order to specify which are the regions that we are interested in to exclude, and mainly focus on the genome duplication effects that effect that we can find in fishes' genomes. So in the case of ENSEMBL, you can see here a region from rainbow trout. We can have from ENSEMBL a file where we have specified different genome repeats that are found through the genome, like, but this file or these annotations are reporting like a lot of different types of duplicates, right? They can be like long tandem duplicates, short repeats, but not all of them will be necessarily highly mappable in our experiments, right? So this is why what we did was to design a pipeline that is mainly based on the experimental data that we have, right? So what we did was to take the different control ChIP-seq experiments we had, we processed them identifying peaks there with GenRich, and then we combined these peaks in order to obtain a set of consensus peaks. So you can see here, these are different peaks identified in different control samples, right? And then we overlap these consensus peaks with the ENSEMBL genome repeats in order to identify which of them were annotated. So basically at the end, we ended just masking the regions that are highly mappable in our experiments, right? So in this case, this region, instead of having all these repeats marked, we only have these three, right? So this allowed us to have less fragmented peaks and also to retrieve more information from different genomic regions that otherwise will be fully masked. Another improvement that we did in our primary analysis pipeline was the introduction of gapped peak tracks for the case of the histone marks that are characterized for broad marks, like H3K4 monomethylation, the mark that is related to active enhancers. So in the case of this pipeline, what we do is like to perform two different peak calling tasks. The difference between them is basically different values for some parameters that are involved in the peak calling process. So we are detecting narrow peaks and broad peaks. You can see here an illustration of a narrow peak tracks and a broad peak tracks. So then we combine these two tracks in order to obtain a gapped peaks track, that is the one that is here in pink, where we report the broad peaks, that are overlapping with narrow regions. And we highlight what are the narrow regions that are involved. So when you are retrieving information from our browser, you will obtain, for this type of histone marks, this type of tracks. After I mean, the aim of the primary analysis pipeline is producing high-quality data that will be used in our regulatory annotation pipeline, right? But not all of the data that we produce in the primary analysis pipeline is later used. Before doing that, we have a quality check step in which we select what are the experiments that we consider high quality for our further analysis. We analyze different statistics, like the percentage of exclusive peaks per epigenomes, overlapping of the peaks with the ensemble repeats that we didn't exclude in our masks, the number of peaks that we obtain, the overlapping between ATAC-seq and ChIP-seq peaks, and based on different statistics, we decide how many of the experiments we produce in our pipeline will be downstream used. In our regulatory annotation pipeline, we have two steps. The first one is the identification of the regulatory features. This was improved in our database for adapting to the AQUA FAANG project. I will try to explain briefly how it works. So basically, we start combining the open chromatin sequencing peaks that we detected. In the case of AQUA FAANG, we just have ATAC-seq, but we could also incorporate DNA-seq peaks. We combine the peaks across all the different experiments we have for a determined species or assembly. For defining a union peak set, this set is then compared or integrated with the annotation of TSS that we retrieve from ENSEMBL. And then we define promoters in a broad way, like calling the regions from these union peaks that are around windows that we define, that are falling, in windows that we define around the TSS. So after this step, we have the set of our promoters, and then we have a set of candidate open chromatins. Then we use the information from these two histone marks that are related to active enhancers for identifying which of these candidate open chromatins will be enhancers and which will be open chromatin regions. So we detect how many of these are overlapping with ATAC-seq peaks, let's say, for identifying which are representing active sites. And then we combine the candidate open chromatin regions with annotations of ENSEMBL exons for identifying the sets of enhancers and open chromatin regions. So at the end, we will have three different types of regulatory features: promoters, enhancers, and open chromatin regions. The next step is to take this annotation and per each epigenome, let's say one epigenome can be one tissue imager style male, let's say brain female imager, and we overlap the open chromatin seq peaks that we have for this epigenome with the different features in order to identify which of them are active or inactive, right? So I have an example of a genomic region here for turbot. You can see here there is a gene annotated. Our regulatory build is reporting 4 features here. The 2 ones that are in gray are open chromatin. The red one is a promoter, and the other one is an enhancer. And then I also added the epigenomic activity that we found for the 6 developmental stages for the turbot embryos. And they are like sorted by more immature to mature, right? So as you can see here, the first open chromatin region is always inactive, and the second one is always active. And in the case of the promoter and the enhancer, both are inactive in the first 2 developmental stages, and then they become active, right? So this is kind of the analysis that you can do with this epigenomic activity. I tried to summarize here the number of experiments that we analyzed for our ENSEMBL 111 release that hopefully will be alive next month. You can see here the number of experiments for the 5 species for which we will be able to annotate regulatory features and the different types of experiments we analyze. And you can appreciate in the darker bars what is the number of experiments that we effectively used after performing the quality control step, right? It's worth to mention that our first release that included regulatory element in fish species was 110. That is the release that is currently alive. So it's the one that you will be able to access if you enter now to our genome browser. In this case, I have here summarized the number of features that we released in this release and in the one that will be alive soon. So for ENSEMBL 110, we included Atlantic salmon, sea bass, and turbot. And at that moment, we were just able to report promoters and open chromatin regions. In our new release, we improved the annotations for these species because we incorporated more experiments, and we also added the annotation for enhancers. And we are also releasing regulatory features for rainbow trout and for common carp. I want to say thank you to all the ENSEMBL regulation team. We are all summarized here. Of course, to all the ENSEMBL team and to all our funders. And thank you for being here, for listening to me. And that's it from me.